Thank you very much, Irvin.
Thank you.
So now we have Francesca Ruscitti from Chiesi Farmaceutici. She's a senior scientist in Chiesi, working on preclinical research and target identification. So I think now we have the chance to see that perspective from, single cell from a different perspective. The industry academy. Maybe you want to take. Up to you. Either you stand and use the mic or. You decide.
I think. Can everybody hear me? Okay.
You can use this.
Yes.
Up to you. Either you stand and use the mic.
No. Oh, yes.
Yeah.
Okay, fine. That. Okay. So first, I would like to thank you, the organizers, for inviting me today and the opportunity to give this speech. So just a few words. I've been working in Chiesi since 2012, and in the I'm so awesome things, I joined the Target Innovation Department. And that's why today I'm going to talk about our approach in support to target identification. So this is the agenda. I will spend just one slide explaining that we are facing very complex diseases nowadays. And then I will move straight to the core of the presentation, talking about the target identification and validation mission in Chiesi. And then the concept of the right target and that we have. And then I will touch the point of the value of single-cell omics in drug discovery. And since Chiesi is a leader company in a respiratory field, I will just give an example of the strategy to target one of our disease of interest, IPF, idiopathic pulmonary fibrosis. And then I will conclude my presentation. Basically, just few words, but the most important problems in the healthcare today is that many patients do not respond to drug treatment way. And this is closely related to another problem, the increasing costs and difficulties in drug development. So one of the drivers of this high rate of failure is suggested by GWAS studies, basically which identify increasing numbers of genetic variants of the same disease. And moreover, this is also suggested by single-cell RNA-seq studies, which basically have shown that altered expression of thousands of genes are present in different cell types. All these things just to say that we are fighting very complex diseases based on genetics, environment, and lifestyles. So the drug discovery process is a long and risky way. We always start from an unmet medical need in order to find a good target. So that's our mission, the target identification and validation. You can see this pink box that cover 1-3 years of work in order to find at least one drug candidate to clinical phases and to have one approved drug on the market. So basically, the target identification and validation is an essential point in order to build a robust pipeline for the company. And our strategic role is, of course, to indicate the most promising path for drug development. And I would like to stress the point that our aim is really to find the right biological target. Not only this, so our mission is not only to validate target, but it's and have the same importance to devalidate them if we don't believe that going to be the right one. So target identification is a critical step for drug development. And I would like to introduce the concept of the drug attrition rate in clinical trials. That is a huge problem that is facing discovery. And basically, this is due to an inadequate preclinical target validation package. So on the right side of the slide, you can see this teeny tiny stone, and we are this stone. So you can easily understand how choosing the right biological target is the critical primary decision for the development of new drugs. There are key challenges. So the drug development Oh, yeah.
Sharing was off.
Oh, I didn't realize. Sorry.
No, no. Sorry for that.
Sorry. So the development process has this high degree of uncertain success, due, of course, also to the identification that is a critical steps, and also moving to the clinical with the unknown mechanism of action of drugs may set the stage of failure. And of course, we are also facing the problem of patient heterogeneity, as I said before. So even high quality candidates fail if they have the wrong target. And the need is that appropriate target identification and validation increase success in drug discovery. Picture taken from a Nature paper from 2015, basically where an analysis of 4 big worldwide pharma has been made, and they demonstrated how the reason for new drug failure in clinical trials is the lack of efficacy in phase 2. So how to increase success discovery? To make a long story short, I will briefly touch this point that basically are the answer to what makes a good target. We always have to look at the clinical needs, then the patient population. If, for example, there is a stratification of patients, we have to look at commercial needs, so the competitive environments and the market opportunities, and of course, do all the assessment in charge of the target identification and validation. And look at, of course, at the IP, so if there is space for new therapy. And of course, if in the literature are in vivo models for the experimental validation. So the key factors for successful target identification are first to understand the molecular mechanism of the disease, identify the therapeutic target that is relevant for that disease, and at least demonstrate the target is relevant to the disease mechanism, so with models and technologies. So you can understand here that the target identification validation is a multifunctional process. I cannot go in details of all of them, but they include a multi-omics approach in order to connect genotype with phenotype. So in the era of precision medicine, a multi-omics approach enabled the integration of data from diverse omic platforms that you can see in this circle. I put them in a circle because it's important to integrate into a system biology approach. And the advantages are clear, I guess, to everyone, because of one of these is the driving rapid improvements in the quality, quantity, and cost of methods for drug discovery research. And then, of course, another advantage is that they increase a robust discovery, development, and linkage of targets to drug candidates. And they allow a fully understanding of biology deep down to the level. So I stole this slide to a webinar from 10x Genetics, because it was very fancy. And I guess all of you are familiar with this concept that we have our samples are complex tissue. and that with bulk analysis, the heterogeneity is masked. So the value of single-cell omics and also the possibility to do also study of spatial location brings some advantages. For example, the possibility to look in the function of cells and their context, discovering new cell types and state, and also for the subpopulation, the new subpopulation detection in heterogeneous samples and the spatial position, and of course, deeper understanding of the pathways related to. So I will summarize here in 3 slides the work of years, just to give you an example on our strategy to targeting one of our disease of interest, that is idiopathic pulmonary fibrosis. So this is a lethal disease. This is a chronic, progressive, and basically it's a fatal disorder with an unknown pathophysiology. And it is characterized by scarring of the lumps and extracellular matrix and collagen deposition, and a progressive loss of function for patients. So only 2 drugs are on the market. They are nintedanib and pirfenidone. They are the standard of care. So and they only are able to slowly, not to progress the disease, not to progress. So there is basically no cure for IPF right now, and that's why this represents for us a high unmet medical need. I put this picture from this very nice paper, not to confuse you, but just to give you the idea that IPF is a very complex disease. It involves multiple cells types like fibroblasts, immune cells, epithelial cells, and pathways. Not only that, but moreover, subpopulation of patients have also genetic factors that are the cause of the disease. So how single-cell studies helped in right target for us? This is again taken from this paper, this very recent, of a single-cell transcriptomic studies that basically have revealed changes in epithelial cell population with a dramatic shift from alveolar cells to bronchial epithelial cells. And not only this, but other paper like the one published by Adams in 2020 found out a new distinct, transcriptional distinct population of cells named aberrant basaloid cells. So this just to say that single cells studies not only find out new subpopulation cells, but also states or differences in the presence of this subpopulation, and helped us to find out which cells are related to the pathophysiology of the disease. This is just to make again an example. There is no way to see a cellular breakdown on a bulk analysis, but we are able to see difference in expression of whatever, proteins, mRNA, in singular population. For example, the cells we are interested in, like epithelial cells or aberrant basaloid cells, this new subpopulation. So our strategy is cell type focused. So first, we are gonna find out cell types associated to the disease. Could be everything. I cannot, go much in details, but this is basically the steps we're going to do. So secondly, we are going to find out altered pathways with also the bioinformatics support of our collaborators with pathway enrichment analysis or master regulators analysis. And then third, in order to find out a list of genes belonging to altered pathway or specific cells relevant for the disease. We are going to do a prioritization of these genes based also on integrated analysis, for example, GWAS evidences. So the final aim, of course, is to find out new targets, but also new pathophysiological insights of the disease. This is my last slides, is to share with you another project that basically is to, again, the aim is, as you understand, is always the same, to identify new relevant genes for IPF. And basically is the project with our accelerator to build a web data repository in which we can integrate data coming from single-cell transcriptomic data sets or bulk transcriptomic data sets, see what studies, proteomics and experimental coming from animal data. And with ad hoc algorithms for gene scoring and prioritization, we are going to prioritize targets for the disease of interest. So to conclude, I want to again stress the point that first, choosing the right biological target is the primary for the development of new drugs. And then also that appropriate target identification and validation increase success in drug discovery. So we have responsibilities. Then single-cell multi-omic technologies really have already contributed to the identification of novel disease biomarkers, cellular subset, targets, therapeutic or diagnostic ones, and many of which would have been undetectable by bulk analysis. And finally, an integrated analysis of omics data significantly impact optimizing the target identification in order to find out new relevant targets in complex human disease. So I would like to thank you for your attention, and the Chiesi team, the target identification department, and our collaborators from the University of Parma.
Thank you very much. Very interesting talk. Again, we have time for a few questions. Actually, I do have a question, but like a curiosity. Maybe I'm wrong, but as far as I understood from your talk, in principle, of course, you should aim at giving the right drug, not just to the right patient, but to the right cell of a right patient.
Yeah.
I don't think it's possible, but what you meant?
Yeah, but the point is that, of course, a lot of pathways and so different cell types are involved. And so, yeah, I'm not talking about, the right cell. That the point is the right target, possibly expressing as possible to the right disease, for the right disease to the right patient. So the three concepts are these. So we are targeting, specific subcellular population if they want, if we know that there is a good chance to target this cell and is relevant for the disease.
That might be the next century challenge. I mean, from the technology standpoint. Of course,
in IPF, all the therapies are also based on, combo therapies. So this is one way, but of course, the idea is also to give a combination of drugs for these complex diseases.
Right. Thanks.
Thank you.
Maybe can I ask a question? Sure. It's actually more of a business question more than science.
Oh, okay.
But I think it could be interesting, maybe especially for the younger population. So I know many people, many friends who now are switching from academia to industry, including at the professional level. Like they are very much attracted to big pharma in the United States. I guess it's true in Italy as well. Although I don't know basically anyone who moves to industry and works on single cells. So is it something that is like peculiar of Chiesi, that you guys are ahead of everyone else? Or like, is it a common practice?
I guess maybe it's something that is happening in these years. So probably the answer is more in the next years, maybe a lot of people will work on this, on this in this way. Yeah.
Yeah, but you guys are ahead of time because I don't know any of my colleagues who do that.
Okay.